Welcome to the conference. Today I will be presenting my abstract, Protocol Description, Biotechnologies for Space Exploring Nutrient-Rich Organisms for Closed-Loop Life Support. I'm Sara Zafirovska, a molecular biology and genetics student at the Faculty of Natural Sciences and Mathematics, University of St. Cyril and Methodius, Skopje. Kombucha and spirulina offer unique advantages for long-duration space missions. Spirulina is a highly nutritious microalgae, rich in proteins, vitamins, and antioxidants, while kombucha provides probiotics that support gut health. Both are resilient, can be cultivated in confined environments, and require minimal resources, making them ideal for closed-loop life support systems. This presentation outlines the experimental protocols used during the Kratos mission to study kombucha and spirulina under space-like conditions. The experiments were designed to evaluate the fermentation of kombucha under different conditions and the use of kombucha SCOBY as a growth medium for spirulina. Now I will present the protocol to conduct and to track the fermentative progression and quality of kombucha in simulated vacuum chamber conditions. For the preparation of the kombucha samples, 600 milliliters of distilled water were first heated. Then 3 green tea bags were infused to create the base solution. Once the tea was ready, 2.3 tablespoons of white granulated sugar were added and stirred until fully dissolved. After that, the sweetened tea was left to cool down to room temperature before inoculation to prevent damages of the microorganisms. Once cooled, 70 milliliters of the starter culture taken from the habitat kombucha jar were added to the solution and mixed gently. The mixture was then distributed evenly across 4 vessels, and approximately 2.5 grams of fresh SCOBY were placed into each one of them. These prepared samples were then ready for incubation and further observation. 4 experimental setups included 1 sealed jar, 1 open jar covered with sterile gauze, 1 vacuum-simulated environment created by the crew members using a plastic bottle, and 1 control sample in a plastic bottle closed but without vacuum. This arrangement allows us to observe how different atmospheric and containment conditions influence kombucha growth and fermentation. During incubation, pH was tracked using universal pH paper, and visual observations were made to record bubble formation and pellicle development as indicators of microbial activity and fermentation process and progress. Now we will present a protocol for the spirulina cultivation using kombucha-based media. For the spirulina cultures, a kombucha SCOBY was blended with distilled water to create the base medium. 3 dilution ratios were taken, then prepared 2:1, 1:1, and 1:2 of water to SCOBY. These media were alkalized using 0.1 molar sodium hydroxide to achieve target pH ranges from 6 to 8.9, 9, 10, or 11. A standard spirulina medium prepared by dissolving 2.1 grams of powdered medium in 100 milliliters of distilled water served as the control for a comparison. As well, the clear kombucha SCOBY medium was alkalized as well. To prepare the samples, we used Falcon tubes. In the first tube was the control tube, and we added 40 milliliters spirulina medium. Tube 2 contained 20 ml spirulina medium plus 20 ml kombucha medium in ratio 2:1. Tube 3 contained 20 ml spirulina medium plus 20 ml kombucha medium 1:1 ratio. And Tube 4 contained 20 ml spirulina medium plus 20 ml kombucha medium 1:2 ratio. And Tube 5 contained only kombucha SCOBY medium 40 milliliters that was previously alkalized, and all of the mediums were inoculated with 4 milliliters of spirulina. The cultures were gently agitated every 2 hours during waking hours to maintain even mixing and prevent the cells from settling at the bottom of the containers. This intermediate agitation helped ensure that all parts of the cultures received uniform exposure to nutrients and lights. To simulate a natural growth environment, a 16 to 8-hour light-dark cycle was applied, providing periods of illumination for photosynthesis, followed by darkness for rest and recovery. These conditions promoted healthy growth, minimized stress, and supported consistent metabolic activity throughout the cultivational period. By carefully combining gentle agitation with controlled light cycle, we were able to closely mimic natural environment while maintaining optimal culture conditions. A lux meter was used to measure light absorbance in the cultures, serving as a proxy for biomass accumulation. The culture tubes were positioned at a fixed distance from a constant light source to ensure all the samples received uniform illumination during measurement. Increased light absorbance was interpreted as an indicator of biomass growth, allowing us to monitor the culture's development over time. To maintain consistency and accuracy, the tubes were carefully handled and movement was minimized during each measurement. Repeated measurements under these controlled conditions provided reliable data on growth trends, enabling precise comparisons between different media and experimental conditions. This approach ensured that observed changes in light absorbance reflected true variations in biomass rather than experimental artifacts. These experiments demonstrate the potential of kombucha spirulina as sustainable, adaptable biological resources for long-duration space missions. This presentation focuses on the protocols used to monitor microbial interactions and growth under simulated space-like conditions, and the actual results and findings will be addressed and presented further on, supporting future biotechnological applications in extraterrestrial habitats. Special thanks to the Astronaut Training Center and to the mission crew Kratos. Thank you for your attention.